All right, so good morning, officially, good morning. Today, I'd like to go over chapter 11, called Controlling Physical and Chemical Control of Microbes. But before we get started, I did want to ask if anyone had any leftover questions from chapter 7 that they wanted to ask before I got deep into 11. Yes, no, anybody, anything? No, we're all good. I'm hoping also that you guys saw the announcement. I am moving this next quiz. So I believe in this class, we're up to lecture quiz 3. I am going to be moving it from Tuesday to Thursday to give you a little bit of extra time. The quiz is next week. It's not scheduled for this week, not tomorrow, but I am planning on moving that quiz from Tuesday, which we normally have our quizzes on Tuesday, to Thursday to give you a little bit more time for 7, the little bit of 3 that we talked about, 11 we're going to go over today, and then chapter 12. You'll notice, Chris, can you put yourself on mute? There you go. You'll notice that the connects are up for 7, that little bit of 3 and 11, and I have not put them up for 12 yet. I need to adjust the outline for 12 a little bit, and then I'll be putting that up. Please, please, please pay close attention to both our Zoom meeting for 12, which will be next week on Monday, and as well as the outlines for 12, because you are not required to learn the names of specific antibiotics and their modes of action. So please don't waste a lot of time doing that beforehand, because I'm not going to ask you those on the quiz at all. So today then, I would like to pick up with chapter 11, and it's pretty straightforward. There really is, it's a lot of memorizing. I think the best picture, when I was looking through these, I did download the PowerPoints for this chapter, and they're almost all words, because there aren't very many illustrations that go with this. Again, it's mostly just memorizing. Yes, Hope, for chapter 12, do not study the names of the specific antibiotics. And it's also in the announcement that I posted yesterday. So I think this opening picture here in your book is probably, I like it because it's an easy way to keep everything organized when you're studying, and obviously doesn't have all the details on it, but it does hit all the main points, all the big points, and then there's more, of course, for you to know underneath each one of these. On the handout, as well as in your textbook, you'll notice that the authors go through the physical agents first, then the mechanical removal methods, and then the chemical agents. And that's because the chemical agents take up the bulk of the chapter. There's the most information about those. And so there's very short section on mechanical removal. So they've put physical and mechanical together, and then the chemical agents. And I split it up that way in the Connect as well. So if you take a look at the note outline, you'll see that it basically just follows this pretty much section by section. It just follows all through the physical agents, then the mechanical, and then the chemical agents. And I was just going to mention a few things. These terms, disinfection, sterilization, antisepsis, which is also known as degerming, and decontamination, which is also known as sanitization. You must know detailed definitions of those. I notice on the handout I left them blank on purpose, because I would like you to actually go in and write down those definitions yourself. And let's see, here are my controls. Words, words, words. Okay, this is an important panel here, I believe it's figure 11.2, it shows the relative resistance from the least resistant organisms to the most resistant microorganisms. So if you're looking at it from this on the left-hand side here, or from the bottom to the top, it's least resistant to most, or if you're looking at it as it goes down the diagonal here, it's the most resistant to the least resistant. So prions are pretty much resistant to every form of disinfection and sterilization. They're literally almost impossible to eliminate. Excuse me. Endospores are usually the gold standard. So you'll notice that when you read through the different methods of chemical and physical sterilizing or decontamination, only if you get rid of spores or eliminate spores do we consider that you've actually sterilized something. And then again, in decreasing resistance or easier to kill, okay? It's important that you understand the difference between cidal and static, and that's listed back here. Cide is to kill, obviously static is to inhibit. And then these concerns about the factors that will affect the death rate, I've also listed those for you in your book on the handout, and I've compressed them down to 4 items rather than whatever they have here, 1, 2, 3, 4, 5, 6, 7 different items here. These are really important because it indicates and helps a person, for example, who's in infection control or in a sanitizing department in a hospital. It helps them decide what the best chemical agent or physical agent is going to be based on all of these factors, not just how many bacteria are there or how many microbes are there. And what they're talking about, the nature of the microbes, they're basically saying, I'm going to go back to this previous slide. I think I want to see them all. Yeah. I'm going to go back to this previous slide here, right? They're basically saying, what kind of organisms am I trying to kill? Is it a mixed population? Is it a pure population? Is it one of these more resistant types or one of these least resistant types? So, sorry, I'm just I should have put those in a shorter PowerPoint. And if I have time today, I will just pick out the slides that I'm focusing on and put them in a separate PowerPoint for you. The other concern is the practical concerns. And I thought, here, they come here. Again, it's a short list. It's in your textbook as well on page 292. And remember, I put in on the handout that you want to pay attention to the whole list. This is the relative order of importance, but the whole list needs to be considered, right? So are we going to sterilize the item or can we just disinfect it? Is it going to be reused or permanently discarded? If it's going to be permanently discarded, there are other choices like incineration. If it will be reused, basically, is it plastic? Can it withstand heat, pressure, radiation, or chemicals? Is it a solid? Is it a liquid? How much, and that's not on this list, how much volume do we have that we have to get rid of or sterilize or disinfect? Am I talking about a 4x4 square of countertop, or am I speaking about a whole room, or am I talking about a mattress, right? That, of course, is also a very practical concern. Is it suitable for the given application? I guess that's kind of what I just talked about. Will it penetrate? And is it cost and labor efficient? And is it safe? Those are all, of course, very important concerns. The rest of this just sort of marches through. These are how the agents can affect. There are 4 different targets for antimicrobial agents or physical and chemical control. They're going to disrupt or destroy the cell wall, the cell itself, or the membrane. Are they going to inhibit DNA or RNA, nucleic acids? And a lot of these agents focus on denaturing proteins. It's a very common method. If you break down the proteins, of course, you're going to ruin the enzymes. You're going to ruin the structure of the cell. And then, of course, the cell is no longer going to be able to function. And again, I'm not interested in all of the super specifics of this. I just wanted you to have the idea of the general So then your book goes through this, these large tables are listed for the different types of disinfection and sterilization. And what I wanted you to notice is, why I'm showing you this particular figure as well as the next one. See how this is like a peach or a pink background? Anything in those tables, which are listed, table 11.3, table 11.2.4, and as well as all the chemical table, which I believe is 11.6, anything that has this peach background to it underneath the type, that is something that can actually sterilize. The rest of these, this lighter purple color or the darker purple, they are not sterilization methods. And I think that helps a little bit as you're looking at it, because there's so many of them and it's so confusing. It can be so confusing. I've highlighted all of the important points on the note outline, and I can just simply tell you that what we'll mostly be focused on in the quiz for this section is the parameters of a particular method. For example, you should know that water boils at 100 degrees Celsius and you need to boil things for 30 minutes. What is killed and what the application is. And you can see that that's listed exactly here in the table, right? Useful in the home for disinfection of water, materials for babies, food, bedding, and clothing. And then, of course, the greatest disadvantage for each. And that's pretty much what I highlighted in the note outline. So you guys can just read this and go through it yourselves. You don't really need me to go through it with you. I did want to talk about the autoclave a little bit, or sterilization. And of course, for whatever reason, that picture is not included of the autoclave. So I'm just flipping through here real fast and I don't see it. So in your textbook, there is a nice big picture of an autoclave though. All right, how do I get out of here? Can I just go back to, yeah, this is what I want here. I'm checking to see if they put it at the end, because sometimes they put things they don't include in their own little PowerPoint slideshow. That sometimes they put them at the end, but they didn't. So if you take a look in your textbook, table 11.3, the second page of that actually shows an autoclave that is able to sterilize. And what is important to focus on here is that autoclaves are steam under pressure. So it's boiling water, but by increasing the pressure on the water and steam that is produced, you can increase the temperature at which the water actually boils. So the higher the pressure that you create inside the chamber of the autoclave, the higher the temperature can be of that water boiling. And so if you notice, right, if we do 15 pounds pressure, we're actually getting a boiling point of 121 degrees Celsius rather than 100 degrees Celsius. And this is enough to kill everything, including endospores, okay? These autoclaves are large machines. You can think of them as, it's comparable to a pressure cooker, but they are much larger and they can be increased, I believe, all the way up to like 135 degrees Celsius with increase in pressure. Now notice that it is a lengthy process. This does take several minutes. The holding time is 10 minutes for light loads to 40 minutes for heavy or bulky ones, but the average time is 20 minutes. And what that means is you close this device. I'm gonna stop sharing this with you for a minute. It's a big chamber that has like a door on it that you either wind closed, or now a lot of them are digital and you can just, a door closes up and seals. And the pressure gets built up inside of there. And in order to build up the pressure and then boil the water, because the water coming into the device, into the machine, comes in at room temperature, or it's hot water, but not boiling water. That whole building up process can take an additional 20, 30, 40 minutes. Then the machine has to hold the 121 degrees Celsius at 15 pounds pressure for whatever time you set. And as the panel mentioned, and as I think I mentioned in the note outline as well, the time that you need to hold that temperature and pressure varies depending on how large of a load is in the autoclave, as well as what kinds of items are in there. For example, if you are trying to sterilize liquid, that takes about 40 minutes holding time. If you're trying to sterilize metal items like surgical instruments, that also takes 40 to 50 minutes. If you're sterilizing paper, like glass syringes or plastics that can be autoclaved and they're wrapped in a paper envelope that's sealed, then that, and it's a light load, meaning there's only 15 or 20 in there and there's a lot of space around them, that only may take 10 minutes of holding time. So it varies depending on how much material you're putting in and also how contaminated it is. Back in the Stone Ages, when I was an undergraduate student, I had work study, and my job was to take all of the waste after the students had grown their cultures and autoclave it prior to disposing of it, because we actually used glass petri dishes. But then I had to wash out the tops and bottoms, dry, sterilize again, empty, before I could put fresh media in them. So sterilizing those big bags full of media that was highly contaminated with bacteria, that took a lot longer than just sterilizing the empty glass petri dishes after I had them all cleaned out. So there are tables, and if you were in the business of doing this, of course, you would have access to all of that information. And some of it is actually trial and error. In our own lab area at Camden County, we have had some, our autoclave is aging. It's 25 years, it's between 20 and 25 years old, and it doesn't function as well as it used to. And we've learned fairly recently that in order to keep down the mold spores, we do need to autoclave our media, not for 20 to 25 minutes, which is what we had been doing, but we upped it to 40 minutes to make sure that all those mold spores were killed and we could keep the media fresh longer. So sometimes it's trial and error as well as guidelines that are published. And then, of course, you can also use dry heat, and they show you some dry heat methods, incineration and a dry heat oven. Again, notice that they're not shown in here, but these should have a peach background behind them. If you look in your textbook, you'll see the correct images as well. And then as far as another physical agent would be radiation. And I did want to mention a couple things. So cold temperature does not kill most bacteria, only some. It merely slows down the activity. And even if you freeze them, deep freeze them, or lyophilize them, which is combining freezing and with a vacuum, you suck the water out so that ice crystals aren't formed. Those are more preservation methods than actually can be used to kill bacteria or viruses or fungus. Radiation, I did want to mention, highlight a little bit as another physical method. We have gamma rays, X-rays, and ultraviolet radiation. And both the gamma rays, the ionizing radiation, the gamma rays and X-rays, these can penetrate very deeply. And again, this should be a peach background behind there. And they're very penetrating, as well as breaking apart DNA and proteins, usually damaging the DNA beyond repair. Non-ionizing radiation, such as ultraviolet light, is a better method for liquids as well as surfaces. I think there's a picture of, no, I didn't put that in there either. In your textbook, again, there's a picture of food being irradiated, and there's also a picture of like an operating room being irradiated. The food would be irradiated with ionizing radiation, the operating room with ultraviolet light. I did want to highlight the particular damage that ultraviolet light does to cells, right? So it's most lethal. Notice the wavelength here between 240 and 280 nanometers. We usually set the devices at 260 nanometers. A germicidal lamp, notice it's 254, so it's in that neighborhood. It can pass through air, slightly through liquids, but only poorly through solids. And by poorly through solids, we mean anything, literally, quite literally, if you put a piece of paper, a piece of plain paper, in between the UV light source and whatever you're trying to damage, like if my underlying hand here is the bacteria, and this is a piece of wax paper, anything not penetrating, the bacteria will live and grow and thrive underneath. So the trick to using ultraviolet light is you have to have completely clean and all exposed surfaces, meaning it won't penetrate into, like if I was trying to sanitize or decontaminate my calculator with UV light, that would be very difficult because of the depth of the buttons, and it can't actually get inside some of these little nooks and crannies. And another disadvantage of UV light, of course, is that it can damage human cells, right? And this is the manner in which it actually damages both bacterial cells and human health cells. The UV light causes the formation of what we call pyrimidine dimers. And I would remind you that the pyrimidines are thymine and cytosine. We usually refer to them as thymine dimers because that's the more common, because there are more often 2 T's in a row than 2 C's in a row. And notice here, so this is the DNA, right? Notice it doesn't just do much damage to the base pairing itself, but it causes neighboring thymines to form extra bonds. This often causes a bump or a bubble. Let's see, I could probably do it better on the whiteboard. So we've got one chain here, and then when we have 2 T's, 2 thymines, 2 pyrimidines that are next to each other in the DNA, we'll put some A's up here, you get this extra bond that's formed, and what that does is it does actually cause kind of this distortion, and that's a little exaggerated, of the DNA. And then that is a warning for some enzymes, some DNA polymerase enzymes. We call them the SOS enzymes. That's supposed to be SOS. They are DNA repair enzymes, and they come in and they're like, oh no, there's an extra bond here and there's this bubble in the DNA. And so what these enzymes do is they come in and they cut out this section, and then they replace the DNA, and then this is where the damage is done. In the replacement, sometimes they make mistakes. So I'll put this, I should have made this edge black here, right? So the replaced part is just the orange part, right? But that replacement DNA, a lot of times there is a mutation there. So I'll put like a red star. Okay, that's kind of bad, but I'll just put it over here. DNA repair leaves mutations. And then, of course, when this DNA is replicated, right, in the next generation of cells, is that drawn? Right, then that mistake will get amplified over and over again. Okay, so the UV light, I'll make it purple because we usually use that for UV light. So the UV light, this is the purple, is supposed to be the UV light. The UV light causes the formation of the pyrimidine or thymine dimers, and then the repair of that damage is what leads to mutations that can then be permanent. And of course, in a bacteria or a virus, that'd be great because we want them to not be able to replicate. But of course, in our cells, that would be bad because it would cause mutations. And then they briefly mentioned filtration. Filtration can be used for liquids, small amounts of liquids, or air, right? In the case of air, we're going to use a HEPA filter. And in the case of liquids, usually it's a small apparatus like this, and it's a Erlenmeyer flask, and there's a filter paper here. And I liked this picture because it shows you, and I think I referenced it in the notes as well, figure 11.10, 1110, is that small organisms, like there's some bacteria, they're getting trapped on the surface of the filter, and the liquid is passing through the filter. These pores in the filter are too small for microbes to pass through. How small? Between 8 micrometers and 0.02 micrometers. So tiny, tiny, tiny. And here's just showing you some yeast cells, I think, that got trapped on the top of a particular filter. The application of this filtration by liquid usually is on small scale, but some beverages, some blood products, I wrote down serum blood products, milk, beer, water. I was referring particularly to water that is being tested to see that it's has been decontaminated, like in a water treatment facility. And then onto the chemical controls, which are super lengthy. I liked this slide because it listed the desirable qualities of a germicide, right? Things that we would need to consider in order to have a germicide, a chemical disinfectant, or a chemical that can control microbial growth that would be useful on a large scale, right? We want it to be work quickly. We hope for low concentrations. We want it to be soluble and stable. We want it to kill lots of things, right, without toxicity. We want it to be able to penetrate inanimate surfaces like tabletops and countertops and chairs and whatever. We want it to be able to be active in the presence of organic matter. And by organic matter, we mean things like sputum, skin cells, blood, feces, right? Any exudate from people or animals. We want it to be non-corrosive, non-staining, has deodorizing properties because we want it to smell nice, and it has to be cheap and easy to make, right? It's kind of a tall order, right? It's amazing that there are this many different chemicals on the list, and it looks like they don't reproduce the table, but again, I would refer you to the table on pages 305 and 306, and it just goes through the different disinfectants. So we can start with chlorine. Right, and again, I have them all typed out for you on your handout on the second page. So the two halogens are chlorine and iodine that we would consider. Notice again, I'm pointing out what it kills. The disadvantages, it can't be exposed to light, which is why bleach comes in these opaque bottles. And in the laboratory, we make usually fresh bleach solutions every 1 to 2 days because we use a 10% solution, but it's ineffective after about 3 days, even exposed to the minimum amount of indoor light. We need to maintain an alkaline pH, and it doesn't work very well when there's a lot of organic material. Iodine is another halogen. It is, they kill everything. It kills everything as well, but you have to use the right concentration and exposure times. It is a little more effective in organic matter and at various pHs, which makes it a little bit more useful. And they list some applications of iodine or iodine solutions. We would like to mention here at this point that you'll see sometimes iodine referred to as a tincture, and that basically means that it has been, or iodophore, and that means it's been mixed with alcohol. Alcohols, only ethanol and isopropyl alcohol, 70%. And this isn't really true. It's really between about 50 to 95%. We say 70 because that's kind of in the middle. Alcohol at concentrations less than 50% does nothing, and at concentrations of over 95% does nothing. It needs to be mixed with water because water helps with the denaturing of the proteins. There's some lists of applications of alcohols. Oxidizing agents, basically we're talking about hydrogen peroxide, and one other oxidizing agent is called peracetic acid. Again, different concentrations can be sporicidal at high concentrations, but I would say that it requires, I believe, longer amounts of time. Some of the applications of hydrogen peroxide: wound killing, killing bacteria in anaerobic wounds. And then there's also, you can use vaporized hydrogen peroxide or that peracetic acid or ozone to actually disinfect industrial items or larger enclosed spaces. Large industrial items, we mean things like if you had to sanitize electronics or you had to sterilize electronics, right? So to set them up in the space station, you would use hydrogen peroxide because it won't damage the delicate instruments, and you would use it in a vaporized form, not in a liquid form. Or ozone, but ozone has some disadvantages of being, has a high flash point, I think, so that's not good. Then list some surfactants or some detergents, right? Basically, they disrupt the cytoplasmic membrane. They have various effects depending on if they are positively charged or negatively charged. It's mostly, this is the highlighted part here, mainly to use the mechanical removal of microbes. It's more effective when combined with chlorhexidine or iodine. Oh, did I skip phenols? Oh, dang it, I did. Let's go back. I didn't realize that. Phenol. So this was the first major disinfectant chemical that was used. Unfortunately, it's very toxic and has irritating side effects, like irritates the skin, but it can kill just about above 1%. It kills everything. The phenolics are a variation of phenol. And notice it says most viruses, and they are not sporicidal. Their main advantage is this, that they can continue to act in the presence of organic material, but they are too toxic to use as antiseptics. You can use them as disinfectants, but not antiseptics. These triclosan, I wanted to highlight that one. That is a phenolic, so these are chemically modified phenols. This one has been severely limited and banned by the FDA in 2017 because just it was being put into everything, into plastics, and used as a disinfectant, like almost like as a permanent disinfectant because it's nice slow release. It turns out that it was causing bacteria and viruses to become resistant to this compound, as well as antibiotics that had a similar structure, so chemical structure. So that's why most things that have triclosan in them have been banned. Chlorhexidine, to be mentioned, it's a chlorine and 2 phenolic rings. This is germicidal, bactericidal for gram positives and gram negatives, some fungus, viruses, but not spores, but it is mild and has low toxicity, so it has been used in hand washing and disinfectants. All right, so back to the detergents. Most of this is, as I mentioned, is the scrubbing, the forming of bubbles, and the bubbles of the soap actually removing debris from your hands or from the area. Notice that if you do include a germicidal component like chlorhexidine, for example, or alcohol, it can dramatically increase, right, how much bacteria is actually removed from a person's hands, right? So this is with millions of bacteria per liter of the scrub water. And if you use non-germicidal soap, there's lots of bacteria that are still in the wastewater, but notice if you use germicidal soap, that goes down significantly. Heavy metals, not super useful. They're toxic in high quantities, it can cause allergic reactions. But I would say, notice here, silver and mercury are the only 2 that are still being used in germicidal capacity. And that is predominantly in added to dressings and burns, silver sulfidine, and silver ions actually incorporated into plastic and some steel. Here is applications of heavy metals. I don't even think, I can't believe they put mercury on there anymore because I don't think that really is even in use. Aldehydes, there's 3 big ones, glutaraldehyde and OPA, ortho aldehyde, I can't say that very well. These 2 both are much more in use. Formaldehyde or formalin, the 37% aqueous solution, is extremely toxic and is a carcinogen. So it's not used pretty much in everyday practice anymore as far as using as a disinfectant. Glutaraldehyde is amazing because it can be a sterilant. However, It needs at least 3 hours to be sporicidal. It's nice because it's non-corrosive and it does remain potent in the presence of organic material. OPA, notice, less irritating, but does not destroy spores, and it stains proteins, which can be proteins in your clothing or proteins like on your skin. It's still listed in embalming fluid, right? And then there are some gases that can be produced for chemical sterilization. Notice the ethylene oxide here, we call it a chemiclave. It's actually like an autoclave, but it has to have special filters on it to vacate the leftover ethylene oxide because it's a poison and it's a carcinogen. And chlorine dioxide, and notice this is used for drinking water, wastewater, food processing equipment, and medical waste. And it was the vapor that was actually used to clean the offices after those anthrax attacks in 2001. And last but not least, acids and alkalis. These are naturally occurring organic acids, and we use them in food preservation. And here's a little short list here. And if you look on something like a loaf of bread, for example, you would see perhaps some of these acids included, or like deli meat sometimes has these acids in them. And while they inhibit, they're all bacteriostatic, they will not kill bacteria and fungus, but they will limit the amount of growth. And that's the end of what I'm going to